Hello, everyone. Welcome back to our AP Bio session. I'm so excited to see you all back here. And we've got some like amazing chatter going on, and I'm so impressed with the participation, and I'm excited to get started with you. Today's session will be on metabolism. So we're going to talk about photosynthesis, we're going to talk about respiration, both oxidative and anaerobic. So great question. I know that the College Board is offering some reviews and they are starting tomorrow, but I did notice on the schedule that they're covering content that is not tested. Now, I feel like what they're trying to do is round out your education so that the stuff that you don't get a chance to probably review in school are going to be offered as a completion of, of the AP Bio content. But it is, as you guys all notice, it is not actually on the test itself. So, and so, yeah, I'm not really sure. And yeah, today's lesson is going to be testable. And I try to really make sure that, we are at the level of guesswork at this point without any more notice from the College Board. They are releasing more details on April 3rd. The only thing we know is that it is at home. It is going to be free response, and it is going to be 45 minutes long. And they're offering 2 administrations of this. And so I believe the May one is still going ahead, but again, more details will come April 3rd. Today's lesson will be covering content you do need to know for the AP exam. So a little bit earlier, somebody was asking about the units. Oh, and then there was a question about human systems and plants. Plants are still testable. Human systems are not. Human systems have been that was released from the start of this year's administration. Earlier in the school year, they did say that they're not testing human systems anymore. So yeah, so I mean, other than that, it is the same content. Now, the issue with the swap from having no multiple choice questions and doing only free response is that it's kind of like a different way of knowing the content, right? Recognition and recognizing a term and knowing that that is like a certain kind of content is a different skill than recalling and having to like think about facts and writing stuff down. So I'm going to kind of like flex a little bit of a different part of your brain. At this point, you've probably done a lot of multiple choice questions. So the questions that we're going to do in this session are going to be only free response, but we are still going to cover some of the background content that is necessary to be able to understand the prompts and the stimuluses, okay? So I'm going to go ahead and get started. Okay, so let me know when you can see my big purple screen. It says AP Biology, purpley blue with lines all over it. And when everyone is an okay to go, then we will get started with our session together. So metabolism is the topic of today. Very, very broad, so many details that you don't need to know. And so I think the focus of this session is going to be more on the bigger picture, the application of some of the foundational principles behind metabolism, rather than all of the nitty-gritty individual details in there, which, to be honest, isn't something that is even the College Board does say that you don't have to know all the details. Daniel, excellent, thank you. And it's just not as interesting, honestly. And you'll see from the stimuluses today that, you don't really need to know as much content as you think. Yay, okay, Aslan, we're ready to go, let's go. So the objectives of this lesson, we are going to tackle questions in photosynthesis, anaerobic metabolism, and aerobic metabolism. Excellent, okay, so I know that we have a fair number of students tuning in for the first time today. And I did talk a little bit yesterday about the tips for free response questions. I'll just throw this on, right? You can always come back and rewatch these sessions later on. So remember, full sentences, explain scientific terms, make sure you're basing things on facts and not what you feel or like. And then start with the questions you're most comfortable with. So be flexible. It's okay. Nobody is saying that you have to do the questions in the order. So grab your easy points and state what you can and don't contradict yourself. So that's like a big old summary, but feel free to, take a look back later on if you want more tips about writing those free response questions. Now, what I did today is, instead of multi-part larger stimuluses, I decided to condense it down and ask just one question per stimulus. It's still free response. I feel like, if I was to kind of make a wild guess about the kind of free response questions you're going to get. It is much more likely, I think, just so that they can have a broader range of the topics that you study, to do shorter stimuluses and then 1 or 2 questions per stimulus, rather than those giant honking like page-long ones with like multiple pieces of data. So that's essentially what I did today. Is I just did 1 stimulus per question. And then we'll talk about some of the content underlying them. Okay, so like we've done in all the sessions for AP Bio, I'm always going to give you time to read and think it through first. And so this is our first prompt. And so I'm going to give you, let's do 3 minutes. 3 minutes to read this through. So this is our intro into this, but here's your instructions. I want you to actually give it a go and write down your answer, but try to type it in. But I don't want you to hit enter. I'm going to give you instructions on when to hit enter, and then we're going to see a big flood of things coming in. But I want you to type out your response for how you would normally want to answer this question. And notice there's 2 parts, 2 bolded words, and make sure that you have both of those parts in your response. Make it complete, make it logical. And so I'm going to give you 3 minutes to read through the prompt, think about the content, plan it out, and then type in your response. When I tell you to go, when I come back on cam and tell you to go, then go ahead and hit enter in your response. I'll see you back in 3 minutes. Okay, come on back. And now it is ready. I want you to hit enter and tell me, what did you think about the reason why cancer cells have a higher rate of glycolysis? So all I got to do is, if you're in the process of typing, finish up your typing and hit enter. I'd love to see your responses. And that way I know how to gear my explanation. If it is content, we can talk about the content. If it is like the connection between ideas, we can talk about that as well. And so go ahead and throw in your responses, and we'll take a look and see how you guys all did. This is exciting. So yeah, Warburg effect. Whoa, look at this go. Nice paragraphs, team. Nice, okay. Yeah, you guys are all on the right track. I'm impressed. I'm impressed. Did you guys all know that cancer cells do this? That if you know of somebody who is suffering from cancer, they're very often kind of like they're thin. Very often they're quite thin, and all the food that they eat often goes not towards nutrition of their body, but it goes to fueling the cancer. And this is actually the metabolic reason why cancers work the way they do, and why they take all of the food and all of the energy we do from breaking down that food. So nice. Okay, so you know what I want from you all is, if you've had a chance to type something in, I want you to go ahead and tell me more specifically, how much ATP does glycolysis make? When you are breaking down glucose and you think about the net result of glucose into pyruvate, and then, you know, pyruvate then gets anaerobically fermented into lactic acid, how many net ATP molecules do you make under the process of glycolysis? So you guys are all on the right track. I do want to make sure that you're supporting this a little bit more with actual facts and numbers, but you all have the right reasoning. So I'm impressed. Well done. Well done. So what do you think? Let's see. So glucose. Let's see. Glucose is a 6-carbon sugar. Gets broken down into 2 pyruvate molecules. So these are 3 carbons. Nice, well done. And so you actually, in the process of breaking down glucose into pyruvate, you actually make 4, but then you use 2 ATPs. So no, it's all good. Mistakes are meant to be made here. So the net ATP gain from this is 2. You're right. You got it. Yeah. So it is faster. It doesn't require oxygen for it to happen. It happens in the cytoplasm of your cells. And this is what cancer cells prefer to use, even though they are in the presence of oxygen. This is what they call aerobic glycolysis. It only really happens in cancer cells, really. It, metabolizes 2 galactic acids to essentially regenerate some more electron carriers, some more NAD+, even though it has oxygen around. So it doesn't actually end up going through to the oxidative phosphorylation that happens in your mitochondria. Now, this is 200 times the rate. So the next thing I want you to tell me is how much ATP, generally speaking, how much ATP do you make when you break it all the way down to CO2, when you go through the process of oxidative phosphorylation? So we as eukaryotes, let's be specific, eukaryotes, how many roughly ATP molecules do you produce when you break down glucose all the way to the very end, all the way down to CO2? Okay, so this is net, and then this guy, got our mitochondria, does its thing. We'll talk more about this guy. And then through the process of oxidative phosphorylation, we end up making a bunch of CO2. And how much? Yeah. So, I mean, the payoff and the investment is kind of a glycolysis specific thing, Flora. But I'm talking a little bit further downstream of this. Nice. Yeah. So these are the numbers I'm thinking about. Yeah. So I would say roughly for eukaryotic cells, it's about 36. And that is an approximation based on like which, shuttle that the pyruvate uses in order to get into the mitochondria, and then as well as some of the NAD H's and stuff like that. But yeah, roughly about 36 ATPs you get. Now, if you think about this, ATP is required for your cell to live, regardless of whether you're a cancer cell or whether you're a regular cell. If you think about the reasoning, the effect of what cancer cells are being done with the prompt, cancer cells produce energy at a glycolytic rate up to 200 times that. Why? Because the process by which you produce ATP through glycolysis is like 18 times less efficient, right? 18 times less efficient of making energy compared to, you know, the usual process of oxidative phosphorylation. Nice, yeah. So, and so not only are these cells rapidly dividing, this is like the key characteristic of cancer cells is that they rapidly divide, they start overtaking other parts of your body. It is now not only just like you're using a less efficient process, you go at a much higher rate. So they're energy dense, they're energy hungry, and they do it much faster as a result because they're only using glycolysis. Okay, so Daniel asking a great question. What is the difference between electron transport chain and oxidative phosphorylation? They're not different. They are, oxidative phosphorylation is a process which is using the electron transport chain. Yeah, so great answer, Flora. Electrons move through the electron transport chain. And then how is that powering oxidative phosphorylation? We have a really cool prompt, actually, that's going to go through this, but go ahead and tell me. It's about 36. It could be, it could go down to 32. It is max about 36 for eukaryotes. Yeah. What do you think? What is the thing that the electron transport chain generates that is powering oxidative phosphorylation? So that, remember, if you think about the inner mitochondrial membrane, it's got all those complexes, right? Like that. And then the electrons kind of like move from complex to complex, right? And then it's pushing things out. So anaerobic is less efficient, Allison, anaerobic. Yeah, what gradient am I generating by pushing these electrons from one complex to the other? It is a chemiosmosis. Tell me what chemical am I generating there? Nice, Michael, well done. It is protons. So as I push these electrons over, this is kind of like the water wheel. I'm pushing these protons up, like into, you know, the space. You got it? And then here's the thing. ATP synthase is something that is different. It is different from the ETC. I don't want you to think these are the same thing. ETC is quite different from the ATP synthase. I know it looks like a peanut, right? It kind of does, honestly. And so this guy will take that nice generated gradient and then it'll flow down its concentration gradient. And the process, this is what is powering the generation of the ATP. Nice, proton gradients, ATP synthase. So they are a couple, and it's going to be another cool prompt we're going to see in a little bit. But yeah, they're ETC, electron transport chain, is used in the context of oxidative phosphorylation. Why do we call this oxidative phosphorylation? Well, the final electron carrier down here is oxygen, right? And so this is a final electron acceptor, and it's oxygen, right? So it's in the presence of oxygen, and I'm phosphorylating, so adding an inorganic phosphate to ADP to generate our ATP. That's why it's called oxidative phosphorylation. So Manel is it as they move through the proton gradient, they affect a pump producing ATP. So the pump is essentially the movement of the protons, right? So that's not quite the pump, but the pump is, I mean, I don't know if I'd really call this a pump. Yeah, so it's the pushing, I guess, the pushing of the protons as the electrons are moving through the ETC, and then that proton gradient is being used by the ATP synthase. Yeah, okay, so Mary, great question. Cancer cells are going through glycolysis. They do it at a much faster rate, then they produce more ATP than normal cells. Well, if I use the math, honestly, it kind of does look like they produce more ATP. And it's because cancer cells divide and replicate so rapidly. So they need a lot more energy. And, you know, dividing is an energetically costly process. I'm creating more protein, more DNA, more organelles, more, like, you know, lipids on the outside. So yeah, it is a very energetically costly in order for you to divide. And if you're dividing that quickly, that's why the glycolysis is so much higher. So for two reasons, it's dividing fast, and glycolysis doesn't produce as much energy as aerobic metabolism. Okay, so if you are like, I get your response, I get what you're saying, give me some words to back that up, then here we go. Here's some words. If you wanted to kind of come back and read the sample response, this would be something that I would be looking for if I was you know, looking at the short answer response for this. Use data, use proof to back up your point. You could say that metabolism is less efficient, but I want you to be more specific here. Yeah, Sadie, a lot of the work here is due to the lack of cell control. You got it. Now, because I went down a Google rabbit hole, I for sure. And I was like, why don't they use aerobic metabolism? You know, it's there. They have all mitochondria. How come they don't just use when it makes a lot more ATP? And it has to do with like the specific kinds of cell control, like you mentioned. There's usually issues with the mitochondria and the specific enzymes that trigger this like shift away from aerobic metabolism beyond the scope of AP Bio for sure, but it's always interesting to kind of connect the pieces and help understand the world around us. Okay. Awesome. Okay, so that's exciting. Let's move on to another prompt here. So I'm going to do the same deal. Okay. I want you to read through the prompt, type out your responses, but don't hit enter quite yet. And then when I come back, I'll get you to submit your responses. We'll talk through the science behind it. And yeah, so this one, I'm going to only give you 2 minutes, 30 seconds for this next one. It's a little bit slower. Allison, real quick. So basically, at a faster rate, exactly. Nice summary, Allison. Yeah. So if you can inject in a little bit of like what makes it less efficient, like with actual numbers, like the 2 versus the 36, and that would be a pretty solid response. Nice. Yes, and yeah, sorry. Glucose is converted to lactate during anaerobic respiration. That's the reason why we have lactic acidosis. So that's absolutely correct. Nice job, team. Okay, so 2 minutes, 30 seconds for this next prompt. Go ahead and type in your responses and we'll enter and review the answers together. There we go. Hello, welcome back. I'm so happy. I know that a lot of you guys put in answers and you accidentally typed it in. That's totally fine. But I appreciate the enthusiasm. I really do. So I'm excited to see what you guys all thought. Go ahead and hit enter. Tell me what you thought. What is some evidence that you would be looking for as a scientist to support the idea that this unicellular organism is photosynthetic? So go ahead and hit enter and let me know what is your 2 pieces of evidence. Make sure to pay attention. You don't have to be like super comprehensive. And there are a couple of different ways to approach this. You can support photosynthesis. You can say not photosynthetic and look for other things. So nice, well done. Look at that, well done. Yeah, so you guys all know, a lot about photosynthesis. Yeah, so chloroplast, chlorophyll, oxygen, organelle. Oh, interesting. Okay, okay. I like that. So let's be clear here. Is this necessarily a plant? Is my unicellular organism here necessarily a plant? Is one have to be true for the other have to be true? Like, are all photosynthetic organisms plants, and are all plants go through the process of photosynthesis? I think one of those directions work, but not the other one. So you want to be careful about your proofs. If you are proving something that is more generic, but not necessarily true about the conditions of this question, which is photosynthesis, then it would likely not serve as proper evidence. Yeah, they're not the same thing. Yeah, so be careful. Some people might have made some assumptions here and said for sure it's a plant, but we don't really know that. All we know is that it has to go through photosynthesis. So yeah, it could be bacteria. You got it, well done. Yeah, so if you provided evidence to support the idea it was bacteria, that wouldn't be photosynthesis though, right? That wouldn't be photosynthesis. So you want to make sure you make it photosynthetic specific. So let's talk about some of those reasons. You guys had excellent understanding. So maybe I will type this out. So let's see. And so remember, you want to make sure that you are typing this out in full, complete sentences, right? So no bullet points, no one word answers, right? So the organism will be photosynthetic if there was a photosynthetic membrane or the presence of chloroplast inside the cell. So if we kind of like took a microscope and looked very deeply at the cell itself, you can also support the idea of it's more likely if you've got oxygen kind of immediately present around the organism. So maybe if it was like in some water, you can look for bubbling. That would be something that would be support. And then if you sampled obviously the bubbles and it was oxygen and not like CO2, then that would definitely support the idea of photosynthesis. Nice. Another way to support this would be something like those pigments. So if you analyze the components of this unicellular organism, it had things like chlorophylls or carotenoids, other different kinds of photosynthetic pigments. And feel free to elaborate a little bit more if you think would be helpful, but this would be like 3 different things. Photosynthetic membranes are part of the photosynthesis process, right? So yeah, there's the whole like the Calvin cycle and all that stuff. So those are all like oh man, my words. The stacks, man, I can't remember, but I'm sure it'll come back to me like after the session is done. But yeah, the individual components inside photosynthesis, any one of those components would serve as direct evidence that would be sufficient for answering this particular question. Yeah. I wish I had time to go through the individual parts of the plant cell, I admit, but while the process of photosynthesis is something that you do need to know, you don't need to know a lot of the individual details. Same thing with aerobic metabolism. You should know the overall process, but all the nitty-gritty details don't tend to be as testable. So like, figuring out what you don't know is important in the process of your studying, but don't like linger and go way too deep. You can do that when you get into college as well. Okay. Stroma, yeah, well done. Nice recall, everyone. Nice, excellent. Okay, so we're gonna move on. I've got another cool stimulus for you. This one's got a little bit of data. So just like last time, I am going to give you I'm going to give you 3 minutes, I think. For this next one, same deal though. Type in your answers, don't hit enter, and then we'll review the answers and explanation together. Okay, team, go ahead and submit your answers for this question. I realize that it does refer to a diagram that you've probably seen in a bio lab before, and that's okay. You don't really need the respirator diagram to be able to understand and answer this question. So if you understand what this is referring to, awesome. If you don't, that's okay. You don't really need it for this question. The important components here is about the cellular respiration measurement and the way that it's generating some data. So take a look at what we got. I'm super impressed. Well done. Nice. Okay, so remember to keep on working on those complete sentences and looking for the proof that is provided in the data table to support your conclusions. So nice job. Yeah, okay, so explain the difference. The first part it says, explain the difference. And I would encourage you all, before you head to the answer choice, the question, to take a little bit of time kind of understanding what the data is telling you, right? I can see that we've got time as an independent variable, we've got temperatures, and then we also have germinating versus non-germinating. So a lot of variables here, and they are all measuring oxygen consumption, the usage of oxygen, therefore probably going to be the production of CO2. So if I kind of like overview this, as I increase in time, I'm going to have more oxygen consumed as I kind of consider the differences between germinating versus non-germinating. I have higher rates of oxygen consumption with germination. Okay, and then if I have higher temperature, I also have higher rates of oxygen consumption as well. So nice. Okay, so explain the difference first of all. It says between the non-germinating and germinating. Well, you can clearly see from the table that there's more oxygen being consumed in germinating versus non-germinating. What does germinating mean? What does that mean? When I'm talking about the germination of a seed or a cell or anything, I don't know if I call it a seed cell, but yeah, what does germination mean? Oh, I love the numbers. That's really good. Oh, if you're hitting the limit, I didn't even think of that, Mia. Yeah, go ahead and separate your responses. I apologize. It'd be kind of hard to answer some of these in 200 characters or less. What does germination mean? What are we referring to when we've got seeds that are germinating versus seeds that are not germinating? These are kind of cool little little cotton swab with a syringe, and it's measuring that stuff. Yeah, it's about the development. It's about growing them. Yeah, so if you ever see a germinating seed, you've done those kind of labs in even grade school, elementary school, they got in a little plastic cup. You could see the seed kind of sprouting, right? Yeah, exactly, right? Just as it's starting to grow from the seed. And so a non-germinating seed would be a seed that's a dud. It's not really doing anything. So clearly, if you're growing, you need to use energy. You need to break down that, the energy sources that you've already got stored up, and therefore that's cellular respiration. You got it. Nice. Okay, at least that's a really good full complete sentence that will explain those differences. Nice. Okay, so explaining that difference, first of all, says, I'm just going to copy paste this. There we go. So the first part of this, I would think something like this. This is a little bit wordy. You certainly don't need it as intensive as this one, but at each temperature, the oxygen consumption is higher. Germinating seeds require more energy than the non, so the rate of cellular consumption is higher. And so oxygen consumption, which is indicative of that cellular respiration, will be greater for the seeds that are germinating. Well done. Okay, so describe the effect. I kind of already threw that in there, but as long as you can understand that higher temperature means more germinating, that makes sense to me, right? I feel like a seed doesn't really want to grow at 10 degrees Celsius versus at 25, which is room temperature. It'd be more likely and happy to grow when it is warmer. Okay. What do the non-germinating seeds consume the oxygen for if they're duds? I mean, good question. It's possible, I might be thinking, well, maybe they're not duds. Maybe they are just kind of present. They're not quite ready to germinate. But the act of getting bigger, dividing, sprouting, things like that, definitely consumes a lot more energy than something that is just there. So it's not that it's actively sprouting, but it is still, hopefully down the line it will still survive. So at first, Mia, when I saw that, I was like, oh, it must be just like baseline. But clearly there's a trend of increasing amounts of O2 consumed over time. So it's not quite like they're just dead, but they're just hanging out in background. Yeah. No, I don't think you really need to be more specific about the temperature, as long as you can say that, you know, increase in temperature, increase in amount of O2 consumed. So Zishan, would a temperature too high degrade the enzymes used in cellular respiration? Absolutely, right? That's kind of the reason why, that if you try to, like, what happens if you take, you know, the popcorn kernels? If you take the popcorn kernels and then you put it in your ground and then you expect a corn to like sprout from that, it's not really going to work. Same thing with coffee beans. All those things are cooked, or they've been roasted or baked or something like that. And they're not going to germinate. They're not going to grow into a plant because they're essentially being killed off. So yeah, definitely high amounts of heat will kill off the necessary enzymes and proteins. They're very heat sensitive. Although plants are a little bit harder for sure than we are. Yeah, it's a really good point, and it's kind of like wildly biologically applicable there. Nice. Okay, so the last part is asking about the differences in the oxygen consumption. I could say something like this, right? As the temperature increases, cellular proliferation increases. Oxygen consumption for germinating corn seeds at 20 degrees for 20 minutes would approximately be about 1.4. If you gave a range, that would still be fair, right? All we're doing is looking for, can you look at the right part of the data? Sorry, can you look at the right part of the data? And so 20 degrees would be like right in between here. And so I would think at 20 degrees, right? So 20 degrees would be something fairly close to the 25 that's here. And so some number kind of within these two, maybe a little bit closer to the 1.2 because we're at a slightly lower temperature. Okay, oh, wait, not the time point. So it's this line, but it's a little bit lower than 1.6. So as long as it's above 1.2, below 1.6, you will get the point for that part of the question. Okay. Well done, well done. I'm impressed. Okay, so how are we all feeling about metabolism as it applies in free response questions? Oh, and yeah, we could talk about that. We're not really calculating anything. Yeah, not 0.8. Remember, 0.8 would be probably like too low here. So what you want to do when you're analyzing the numbers for approximating a value, so let me just erase my stuff here. What you want to do is think, okay, well, the question is asking me specifically for, the at 20 degrees. So it's this last line and it's for germinating. So this column and this last line. So this is my frame of reference. So this would be 25 degrees Celsius. It would be 1.6 mils of O2 consumed. So it's the same deal, right, at 20 minutes. And so all you got to do is, well, 20 degrees Celsius at 20 minutes would be something lower than 1.6, but it's not like super, super low. So I wouldn't go as low as 0.8, but I would say something probably above 1.2. That's what we're doing. No calculations here. It's just a rough approximation. And it's really the skill here is asking, do you understand how the data is sorted? And are you able to kind of like predict the effects if we change some of the variables? Nice. Okay, so Manal and Danielle feeling really good about this. We got one last kind of a bigger prompt to go, but it's such a cool problem. And I'm excited to do this one next one with you. Okay, so this one is a little bit of a more challenging problem, but it is also just really cool and directly relevant. So just like last time, I'm going to give you 3 minutes to do this one on your own. But if you find that this one is challenging and hard to do, that's okay. We will walk through it together, and I want you to consider it more of a review of metabolism rather than something that you're going to be stressed about if you can't answer. Okay, so do the best you can. It is based on the content we actually just covered in previous questions, but it is applied in a kind of a new, different manner. So 3 minutes to work through this one. Pay attention to the context of the prompt and the questions, right? You're not having to explain all of it. So 3 minutes for this next one. Okay, team, we're going to break this down step by step. We're going to first approach the top part. We're told that DNP is a weight loss miracle drug. It works so well that it can kill you. Yeah, what does that mean? What does uncoupling mean? Now, I did take a look at the requirements of the AP Bio exam, and it does actually have a section that specifically calls out uncoupling. So I thought this would be a really good opportunity to talk about what coupling and uncoupling means. So excellent. I didn't even tell you, I totally forgot. So if you have answers, go ahead and throw them in and hit enter. Nice. Forgot what uncoupling means. That's okay. We'll talk about it. More O2. Yeah, awesome. I love it. Hyperthermia is abnormal increase in heat. Body can't regulate. Oxidative phosphorylation is unable to occur. Okay, so this is certainly a much more challenging question, I will say. So that's okay. This is where we're going to learn something pretty cool. Okay, so this is actually referring to something I mentioned a little bit earlier in the very first question when we talked about ETC and oxidative phosphorylation. Yeah, it is cellular respiration, but again, to get the points here, you want to make it more specific. What is uncoupled? When you have something that is coupled, it means like it's almost like this is fueling this next step, right? And we're talking about the uncoupling process in oxidative phosphorylation. Really, this is referring to the ETC again. So I'll draw out my nice little membrane here with the complexes, right? And then remember, we talked about the whole electrons going from process to process. In the process of pushing those electrons over, it's going to push out your hydrogen ions. Okay, so I'm going to form that chemiosmotic gradient of that protons in the intermembrane space in the mitochondria. Yeah, it does disrupt something. Now, it's not disrupting the electron transfer chain, but it's disrupting the gradient that's formed here. So this is super cool. It's unfortunate, but it's really cool. So let's talk about this. Remember, and a lot of you guys talking about the technivia are definitely on the mark. Well done. So here we have our ATP synthase, right? And so this is essentially going to allow for the flow, right, of the hydrogen ions down their concentration gradient. This is essentially like a water wheel. Sadie, very nice, cool. It's like a water wheel. So it's essentially, I created a big dam of water in the intermembrane space of the mitochondria. So I've pushed all the water there, and essentially if I let like a little channel go, the water flows down, and that flow of that water, the flow of the protons is what's driving the production of our ATP. So in the process of doing this, we're going to take our ADP and PI and produce ATP. This is how you generate the energy that your body requires in order to survive. Now, an uncoupler is essentially doing something bad to that link between the ETC, right? So if I do this, so there are many mechanisms of uncouplers. Some of them are like they poke holes in the inner mitochondrial membrane. Some of them disrupt by going around the ATP synthase. There are many different kinds of uncouplers. So it's not specifically asking you about the mechanism by how it uncouples, but it's asking you what uncoupling means. So if your answer was something to do with disrupting the proton gradient, then you would be on the right mark. Okay, you were disrupting the proton gradient generated by the ETC. Now, let's say we go back to our analogy of the dam, and if I poke holes in the dam and it's going around the hydroelectric, machine thing, I don't know the term here, and if I've got little streams of water going around the dam, how much energy do I make? Do I make more energy or do I make less energy? If I poke holes in my dam and the water is going around the generator, do I make more energy or do I make less energy? So I've essentially gone it around. Kind of not, I don't know if it's an opposite, TH. I don't know if a coupler would be an opposite of a channel. A channel is essentially an opening. Sometimes it's very specific to something, usually it is in your body. But yeah, channel is just an opening of some sort. So the ATP synthase allowing protons to flow down is an actual channel as well. Yeah, the coupler isn't a macromolecule, team. It is a biochemical process by which we're linking one thing with another. Yeah, I'm totally going to make less energy. I'm going to make less energy, and it's the same deal here. If I poke holes in my dam and I don't make as much protons, well, my ETC is going to run as fast as it can because I'm not making enough energy, but that energy isn't being channeled in through the ATP synthase as well. So yeah, so that's essentially what's going on. So in terms of a correct answer for this part of that first part, you know, I would want something, this is, again, probably way more comprehensive than you need. But again, you can use it as reference later on. You know, it talks about the ETC, creates the gradient, the ATP synthase allows for that gradient to flow down, generating the ATP. And then if you uncouple it, it means it's dissipating the proton gradient, which results in less ATP made. So here's the thing, Allison, the great thing about this problem isn't asking you how or why. It's just telling you that DNP works to uncouple things. So your job in this problem is to recognize what uncoupling means and what coupling means and how the ETC is intimately linked with the ATP synthase, but they are not the same thing. One generates the power, the water, the dam, and the other one uses that dam, that energy, to create the ATP. Yeah, so you need to mention both the ETC and the chemiosmosis with the proton gradient, and then talk about like that's the process of coupling. Okay, so you need to essentially describe the ETC linked with the ATP synthase in order to get that first part of the answer. Exactly. Yeah, let's talk about that, Mia. This is the coolest part. How does that describe, how does that relate or to help explain why you get weight loss? So if I eat glucose, I have like my favorite bag of candy, and then I break it down, but I'm not making as much ATP, then I need to eat more in order for me to sustain myself, to be able to live. So what happens is that instead of like you eating more, which is going to happen for sure, you're also going to start breaking down your fat stores and your glycogen stores. So all the stores in your body are going to start being kept like broken down. And that's why you actually lose weight. Right? Your fat is actually a very efficient energy store. We don't really tend to use it unless we get really hungry, right? And that's what like things like fasting and you know, diets work the way they do. Is if you deprive yourself of nutrient, it's going to start breaking down the fat stores in your body. So if you don't generate as much energy, your body's going to start breaking down those stores, and that's what's going to lead to the weight loss. Tachypnea is actually described in the prompt. It says it's elevated breathing rate. Elevated breathing rate. So if you remember a little bit earlier, I described how this is, sorry, I'm just going to grab a pen, right? So this is the final electron carrier. This is why it's called oxidative phosphorylation, right? O2, right? It grabs my electrons and that then gets converted to water, right? Obviously in the presence of hydrogen. But yeah, oxygen is the final electron acceptor in the ETC, generates a bunch of water. If I have higher rates of ETC going because I've like poked holes in the proton gradient, I'm going to need more oxygen. That's going to result in the higher breathing rate. Okay, so that's the tachypnea. The other one is hyperthermia. So remember in previous sessions, I talked about the importance of root words. We've talked about hypothermia probably pretty often. That's like really low body temperatures. Hyperthermia is essentially the opposite. When you have elevated body temperatures, why would that be the case? Well, you guys have ever been around somebody with a really high metabolic rate? They tend to be like super skinny. They eat ridiculous amounts of food, but it never really sticks to their body. And they've got another characteristic that I don't have this kind of body type. But if you've ever been around somebody who has a high metabolic rate, they're usually quite thin. They eat an enormous amount of food, and they're actually kind of hot all the time. They've got really high body temperatures. So that's essentially what the process is. Hyperthermia is elevated body temperature. The reason why you have an elevated body temperature is because this is an exothermic process. The process of breaking down glucose in the presence of oxygen to produce CO2 and H2O is essentially combustion in your body. Combustion is exothermic. So higher rates of breakdown of macromolecules is always going to generate more energy. Sorry. Exactly, Manel. Yeah, so fat goes up to, you break down your fat to make up for the lack of energy, or you eat more. Sorry, 1 second. So yeah, so what I'm going to do is throw in the rest of this explanation, but you guys did really well. This is a hard prompt. So if you found this challenging, it's totally okay. So I'm going to throw this in. You can go ahead and read that afterwards. But yeah, identifying means what is it? Describing means, sorry, elaborate a little bit more beyond it. And I think getting familiar with the kinds of questions that exist in the prompts, especially the bolded terms, that's an important part of learning how to do this test really well, okay? So free response questions are a bit of a change for a lot of you. Hopefully it's not brand new, you've had a chance to practice this before, but definitely shift your focus towards like explain it in simple terms to people around you, right? Even if you're not very comfortable with writing it down, being able to explain to someone else gives you that mastery level knowledge. So yeah, you're so welcome. I had a lot of fun. What I would love from you all is after the session, throw in some things you'd like to see tomorrow. We're going to do some cell cycle and energetics, so some homeostasis stuff. And if you have like a particular topic or a particular kind of thing that you'd like to see in tomorrow's session, we'd love to hear from you and try to incorporate it in, because these sessions are really for you all. And so I'd love to get some feedback from you all in order to make the sessions more customizable to you. So thank you so much for your time. I will see you back here tomorrow, hopefully, and we're gonna do some cell cycle and homeostasis. Have a great afternoon.